for 504?
Yes, I really wanted to talk about it. It's been it's
Huh?
definitely been a little bit frustrating to see that, I've the BB hasn't really gone up,
Okay.
or CP, but yeah, I have a bunch of questions. Let me see. Okay. So I guess we can just start off with cars. So I mean, I don't think we're gonna I don't want to review cars today because I think the issue is like I felt that it was a lot harder,
Mhm.
but the only thing is um it just I still can't finish 9 passages in a 9 130, right? I guess I still haven't gotten used to all the mapping. Um, I saw myself using mapping like on the first one, but it took me a while and then I got really behind and I kind of, for the most part, I kind of ditched it because I was just really behind. So I don't know, maybe I should set a goal and say, you know what, like this full length I take this week, I'm gonna do maybe like 6 to 7 passages and just set at that and then slowly build my way up, or what do you think? Because yeah, I genuinely can't do 9 right now at least.
Yeah, you said you genuinely can't do 9?
Like if you untimed, yes, I can do 9. But like with the timed conditions, it's really hard for me to do 9 right now.
Got it.
Um, and
And how much do you get through?
it takes me like this time it was kind of bad. It like in the hour and 30, I only did like 5 passages. But last time I did like 6. So,
Wait, you said for the entire t duration, you were only able to do how
I
many?
mean, in a time lot, like when there was like 20 minutes left, I was around passage. Like 20 to 25 minutes, I was around passage 5.
Hmm.
Yeah.
So, um, so l- uh, tell me more about that. So, like, you finish, like, you run out of time on cars, having done how many passages?
Like, yeah, like this time it was barely like 5, to be honest. And then like the last 4, I kind of just rushed through it, to be honest. I rushed through it.
Got it. And let's see, only had time for 5 passages for cars on this most recent test. That's accurate.
Yeah, I mean the last 4, I don't even want to count it because it's like I just honestly I rushed through it. I was not even mapping it. I wasn't really doing it
Got
correctly.
it. And from the previous full length, Blueprint full length 7, was like you ran out of time with how many passages done for that one?
I think it was, yeah, the last 3 I wasn't really able to do. But I did very well on the first like 30 of that half test. But yeah, the last 3 passages I was not really able to do.
Got it. So so the first Blueprint full length 7, you only had time for 6 passages, and then for the latest one, only had time for 5 passages, right?
Yeah.
And what were you using for these?
Jack Weston.
So I thought you ran out of Jack Weston passages.
It was the full length ones that I've been doing.
Got it. Okay. So, all right. Okay. Okay. And for the other sections, timing is okay.
Not really. I was gonna talk to you about that, but I just wanted to start like for cars first. Like I wanted to talk about cars and then we can move on to the other stuff.
Yeah. Tell me about the timing for the other sections.
Yeah, I mean if that's yeah, so that's another issue. So like for CP, right? Um, yeah, so like let's say like the lot this time, by the time I get to 24 minutes left on the exam, a lot of it last 7th and the 8th full length that I took, I still had around 24 to 25 questions left.
So let me see this. If I can do it like this. So let's see.
So wait, let me just I keep let me share my screen. So yeah, so are you ready?
Oh yeah. So I think the timing, I'm just gonna write timing checkpoints would be at question, whoops, question question number 20. Oh, Okay, so question number 20, you should have um let's see. So okay, question number 20, you should have 1 hour left. Oops. Question number 30, should have 45 minutes left. Question number 40, you should have 30 minutes left. So let's say for blueprint full length, let's do like the most recent one, 8. Um, so when you got up to question 20, how much time
Yeah.
did you have left?
I was actually pretty good on that. I think I had like an hour left.
Okay. So now, question 30.
I would say like maybe, yeah, this was bad. I think like at least like 33, 34 minutes. Because by the time I got to 24 questions left, I only had 24 minutes.
And by the time you got up to question 40, how much time do you have left?
Yeah, I would say barely like 18 to 20 minutes left on the test.
Got it. So, okay. So I guess like for that first 20 questions, you're pretty good with timing. But after that, it starts to get kind of shaky with, you know, when you get up to question 30, you are like 12 minutes behind. And once you get to question 40, you're about also like 12 minutes behind, I guess. So, okay, that's something that we can, so that's good to know. So that's something um, that we can work on. Now, you said that this was for CP. Would it be accurate to say that this is for this type of timing, like actuality is true for BB and Psych Soc as well?
Oh, Psych Soc? No. For
Okay.
BB, yeah, the Psych Soc, I finished with like 30 minutes left. I feel like the Blueprint PS seems pretty a lot easier. So,
So this is true for CP and BB, you're saying.
yes.
Okay, so I'm gonna add that in a note. So, um, started running out of time around question 30. Then now, so yeah, so you've been using the checkpoint strategy, but um, what about like the 1.5 minute rule thing?
Um,
Meaning you don't want any questions or problems or whatever to take longer than 1.5 minutes. If it does, then you make an educated guess and you flag it. So yeah.
right. The only issue with that is sometimes since you told me like I should probably stick to reading the passage for CP, like it takes me a bit to like understand that stuff. So by the time I get through, I'm like,
Mhm.
like I get a little, I guess a little bit of tunnel vision. Like, dude, like I just read this. Like, I want to be able to answer this question. So I
Got it.
guess a little bit of tunnel vision, and then I kind of just don't. I guess I just don't keep going.
Yeah, so let's look at your score report for the blueprint test. I think you have it open.
Oh, yeah. Let me see.
Okay. So I see that there's a lot of questions I mean, not including like, the first question of the passage, because that obviously will take longer because it takes into account the time that you spent reading the passage. But I'm seeing a lot of these other ones where you spent longer than 2 minutes for a lot of these passages. And it seems like around passage six is when you started to run out of time. And so the questions that you are answering, you know, passage six onwards, it's taking a lot less time. Um,
Yeah.
so I think that we can improve like the discipline of not letting any question take longer than 1.5 minutes. Like we want to get to the point where your tests show like the
Mm.
maximum amount of time, you know, for these questions, not including like the first question of a passage, to be like around 1.5 minutes here. So I think so you've been following the checkpoint timing strategy like properly. So I think it's a matter of doing the 1.5 minute rule thing.
1.5 minute rule thing. Okay.
Yeah.
I think that's a good thing that you said. Okay, I'll try to I see that's a nice little goal to set. Okay, so trying to be
Yeah,
at 1.5, yeah.
because theoretically, if you don't let any question take longer than a minute and a half, you should get like by the time you go to the or finish your last question for the section, you should have some extra time left over, and that's a great position to be in because that means that you've seen all of the questions and all of the passages so that you can use your own judgment when you hit review flagged to go back and answer the flagged questions. Because by that point, you will have seen everything. So there's no surprise. Like, so what I'm saying is, like, right now, it seems like, okay, right here is when you started to run out of time. And so then you started to, kind of like rush and stuff. And that's fine. But there's no telling like how difficult the rest of these passages could be, and different passages will take different amount of time to like read. In addition to different questions, and different questions will vary in terms of difficulty, therefore vary in terms of how long it takes. So
Mm.
rather than so like, if this happens here, we don't really know what the passages six, seven, eight, nine, and 10, like we don't know how difficult they'll be. Like, because so let's say that, theoretically, right, there's 10 passages, or rather 59 questions. So like let's say whatever, 60 questions. And it's like 95 minutes, I think, something like that. Um, I'll just do like 90 minutes. So basically like 30, or yeah, three goes into this 20 times, or I could just get rid of the zeros. Three goes into this three times. So you see that's 1.5 minutes.
Okay.
Like that's where like the math comes from. Now, theoretically, if you do that and you don't let anything take longer than that, you should have. So basically, what I'm trying to say is that if you follow this rule of not letting any question take longer than a minute and a half, and if it gets to that point where it takes a minute and a half, you make a guess, you flag it, and you move on. And if you do all of this properly, when you finish that last question, and you have some extra time, you're in a position there where you've seen all of the questions, you've seen all of the passages, there's no more surprises, and then you can use your own judgment in which questions that you flagged to review.
Okay.
Okay. So, yeah, so let's try that. Uh,
Okay.
and also, I'm curious about like the types of questions, like the common types of questions that people get, like kind of hung up on, are like the calculate for CP, it's the calculation problems. Is
I
that
mean, I actually, yeah, like I spent the entire day yesterday. I went through the what is it? Yeah, I started to I did this. I sat down and I did all of this. So I'm trying to review all of like all the stuff that I did.
Okay.
Maybe it's good input to for you as well if you want to like go
Mhm.
through it, I guess.
Yeah, yeah. So, for these CP questions, I wonder if there's like I'm just looking through what you have over here. Oh, okay. So this is the math one here, right?
This?
Yeah, it seems like it because you wrote.
Yeah, like there was a value there that I
Yeah.
just I just couldn't find in the passage. And if
So,
I did, I would have probably gotten it because I got it on my blind review. Like I got the answer on my blind review.
so for
So
this type of document, it will also help me to if you for each of these questions input in how long it took you to answer that question.
okay, okay, I'll do that next time. Okay.
Yeah, because like so for
So
instance,
you would be like two, oh my God. Oh, it tells you the, oh shit. Okay. So it tells, okay, so 244, you want me to put that stuff on there?
okay, let's see. Question seven, go back. Question seven. 2 minutes, 23 seconds. So now if you go back to your document here, question seven, this is a math question, right? Couldn't find the concentrations in the passage to convert from. So this took, 2 minutes and 23 seconds. And that often is the case for a lot of people with calculation problems because maybe when you did the first calculation, it didn't give you like an answer that you saw in the choices. And so you would want to try to redo that calculation. But I would say that for calculation problems, you do it once. If it doesn't give you your answer, flag it, guess, make an educated guess, flag it, and move on and come back to it, you know, if you have that extra time left over at the end. So
Okay.
yeah. So okay. It's good. Yeah, I see this is also math. Yeah. So okay.
Oh, there's a code. I found some like some person on YouTube. So they had this whole thing. So I kind of just like been following it.
Yeah.
I mean, I'm noticing a lot is not, it doesn't seem like it's content issue, but the point is like, I'm just slow. As I can, there's no other way to put it. I think I'm just slow in understanding stuff. So yeah, just timing, it just becomes a thing where it's like I can't um put everything. Because like when I did my blind review, I think I got an extra like 10, 12 questions. I forgot what it was
Yeah.
that I understood.
Got it.
Um, but yeah, like it's I guess I just have to, I mean, I'm gonna try to be more aware of a minute and 50 seconds. I mean, sorry, a minute and 30 seconds, and maybe that can help.
Yeah, yeah. And also like so, I say to like read the entire passage, but if you are there's that one exception to that. If it's CP and you're running out of time, as it seems like to be the case, you know, it's okay to read the questions first.
Okay.
Like if it's for CP, and you're running out of time. So like let's say something like this happens, right? Where the 40-minute or the question number 30, right? You're supposed to have 45 minutes left, but for you, question number 30, you had, you know, think like 33 minutes left or something like that. That
Yeah.
that's when you can say, okay, I'm running out of time. So now I'm gonna just read the questions first for CP. That's the only section where you can kind of get away with it. And, it shouldn't be your go-to strategy, but it should be a last resort. And the last resort, like this qualifies as a last resort, meaning, you're running low, you're running uh out of time by that second checkpoint.
Second checkpoint, let me see.
Yeah, question 30 is the second checkpoint.
Question 30. Okay. 45 minutes.
So,
Okay.
like, yeah, the first checkpoint here seems like it was fine. But question 30 is when you started to run out of time. And I would say that's when you can switch gears in the sense that if it's for CP, that is, that you can read the questions first because CP can technically be kind of like pseudo discrete anyway.
Yeah.
This strategy like this wouldn't work for the other sections really. But yeah, it seems like that's something that we can do for CP to improve your timing and your, ability to answer the questions in addition to like the 1.5 minute rule thing. So,
Okay.
yep.
Okay. Um,
Let's go down.
and then like, what's up?
Oh yeah, let's go down to yeah, BB. So, let's see. Seems like for BB,
Like I did okay up to like question 30, and then it just went downhill after that. Um,
so it seems like around here is where it kind of, but I guess
um,
out here as well.
yeah, I think honestly it was around here. 32. I remember this somewhere. I forgot this passage. I was actually reviewing it right now. It was around this passage where I was just,
Mhm.
yeah, I was like, I was so,
Yeah,
I just didn't understand it at that point, and at that point, it just kind of ruined my confidence,
yeah,
and then it just pushed on to the rest of the test, and yeah.
got it, got it. Okay, got it. So okay, now we know what we can do timing wise. And all right, yeah. Why don't we go over some of this stuff?
Okay. And from like at least what you see, what do you think it is? Like at least today with I mean we can review I wanted to really review biochem because I feel like that feels more in my control of doing better on it compared to CP. Um like I was hoping maybe you could also see if like is this really a content issue? Because I'm still having sometimes a tough time to realize. But um let me see. I think there was one passage here. No, wait, let me see. Okay, I think we can do this. But yeah, if you can like let me know too if it's like a content thing. 'Cause I'm still for BB, it's like it's hard for me to understand if it's just an issue with passage analysis. And
Mhm.
I guess I haven't been doing a lot of that recently, but I don't know.
So, is this a passage where you got like a good amount of questions wrong?
Yeah, I got okay, yeah, I got three.
Oh, you
Yeah,
got
four. I got all of them wrong. And
okay.
then I think and then this last one I wasn't even able to attempt it.
Yeah.
I mean, it seemed pretty complicated when I looked at it, so I guess it's something that we both can look through.
Okay. All right. So, have you done your blind review of this yet? Or were you?
Yeah, I did this passage. I did this passage as well. So like I understand some of it for
How
the
was?
most.
Oh, how was it after you did your blind review?
Let me one second. Let me get my iPad real quick.
Sure.
Yeah, I think I only got even after I did blind review. Yeah, I think I only got 49 right on this one.
Okay.
Okay, that's I see that just clicked in my brain that I can also use a blind review to emphasize on what we can go over.
So, okay, let's try this passage out. So, um, I guess I can start it off. So, unesterified long chain fatty acids, LCFAs, are normal metabolic um intermediates, but become toxic within the cell at high levels. Mammalian fatty acid binding proteins, FABPS. um, bind the anionic component um, of these potentially toxic LCFAs in the cytosol and promote their rapid removal in oxidative organelles, the endoplasmic reticulum, and lipid droplets. What are we saying here?
So it seems that these LCFAs, too much of it can become a problem. So we have these binding proteins that seem to bind the anionic component of these LCFAs because fatty acids have that carboxyl group.
Yeah.
And to be able to remove it from the organelle, which promotes the removal of this to prevent that toxicity, I guess.
What do they mean by unesterified?
Unesterified. There's no ester group, so there's no like the carbonyl O to the OR group.
So tell me what that means exactly, like the unesterified.
So I guess in the example, like maybe like we can take it um like a like glycerol phospholipid with its phospholipid backbones. I guess the the glycerol, like that part wouldn't be part of the fatty acid. Like it would just be the carboxyl group. Like wait, let me see if I can try it. Oh, come on. Like it would just be this, right? Like there wouldn't be this R group wouldn't be here, right? If it's unesterified.
Yeah, like a fatty acid, like so tell me the difference between a fatty acid and a lipid.
Okay, so a fatty acid is just a long chain of just with the carb like this O group. There's this carboxyl group, and then just a bunch of like
Would it be the OR though?
no, it wouldn't be the OR group.
What would it be?
It would just I think it would just be this O and then just a negative, right?
Yeah, or an OH, right?
Oh, OH. Okay.
Because it's a fatty acid, right?
Mm-hmm.
So it'll just so this is the carboxylic acid end of it, right?
Yeah.
And then to the left is just that long chain.
Hmm.
What can we add to this to make a fat?
What can we add to this to make a fat?
Hmm.
Um, Not sure.
What did I just draw here?
Glycerol, I think.
Glycerol, right?
Yeah.
So three fatty acids and a glycerol molecule would give you what?
A phospholipid.
Where do you see
Or
phos?
just I guess a lipid because there's no phosphate group yet. Never mind.
Yeah, and
Yeah, glycerol, yeah.
what would that lipid look like?
Yeah, so it would just bind to this, and then we have a fatty acid chain here, and to this, right? Because I think what is it? This lone pair would attack this carbonyl, and then you would have a leaving group of a water molecule.
So you have dehydration synthesis, you're saying?
Dehydration since it's loss of yeah, yeah.
Okay. So your molecule will then look like, and then for the other ones, like this, right?
Mm-hmm.
And you see how we have our ester. there, right?
Yeah.
See that? So
Yeah.
when fats form like a triglyceride with, three fatty acids and then a glycerol molecule, it'll make a like the bond that's made in a fat is an ester bond, right?
Hmm.
Okay. So, okay. So that's good. Okay. So they're saying that yeah, these unesterified long chain fatty acids are normal, metabolic intermediates, but become toxic within the cell at high levels, and FAB-P bind the anionic component. What would that be, the anionic component?
I'll try it again. So probably to the this, right? Sorry for my big negative charge, but yeah, the carboxyl group, I guess.
Yeah, you're talking about that, right?
Yeah.
Good. So, um, so the FAB-P binds to that anionic component of potentially toxic LCFAs in the cytosol and promote their rapid removal in oxidative organelles, um, the endoplasmic reticulum, and lipid droplets, right?
Okay.
What's an oxidative organelle?
Oxidation, beta oxidation, so mitochondria.
Okay.
Oh, and I guess according to this, apparently the peroxisomes, because they can break down
Yep.
hydrogen peroxide.
Yeah, yeah. Because what's happening here is we are in an oxidative organelle, it carries out like a redox oxidation reaction. And yes, the peroxisome is a good example of that. So, okay, good. All right, I'll continue. So, one such protein, L-FAB-P, is highly expressed in the liver and is able to regulate the expression of the L-FAB-P gene, which encodes it. So, yeah, so this protein's very much expressed in the liver and can regulate expression of its own gene. The promoter region of the gene, shown in figure 1, contains response elements involved in fat metabolism, including the peroxisome proliferator activated response element, PPRE. Um, so PPRE is something that is in that promoter region of the gene. Um, which includes response elements that are involved in fat metabolism, like PPRE. Proliferator activated receptor alpha, PPAR alpha, is a receptor protein found in the nucleus that is activated by when bound by a variety of cellular ligands, including LCFAs. Um, okay. Binding of activated PPAR alpha to PPRE has been shown to increase expression of LFAB-P and is required for expression of genes involved in peroxisomal and mitochondrial fatty acid beta oxidation, um, and lipolysis. Additionally, the promoter region contains two sterol response elements, SRE1 and SRE2, along with binding sites for the transcriptional transactivators, CCAAT slash enhancer binding protein, C slash EBP, and activator protein, AP1. Okay, a lot of stuff going on here. So let's see what we can do. Um, so we know that So we could say Fab P is proportional to, or rather, inversely proportional to cytosolic SCFAs because, they help remove the cytosolic LCFAs. And then they're saying that LFAB is highly expressed in the liver and can regulate the LFAB gene. The promoter of this includes things like PPRE, PPAR alpha is a receptor. Okay, so receptor found in the nucleus that is activated when bound by a variety of cellular ligands, binding of activated PPAR alpha to, okay, so let's see, binding of, let's see, PPRE
Hmm.
binding to PPAR alpha causes increase in LFAB expression and is required for expression of genes involved in peroxisomal, okay, say also promote proportional to peroxisomal slash mitochondrial beta oxidation and lipolysis. Um, but it also has SRE1 and SRE2, um, along with binding sites for these transcriptional transactivators. Okay. So then we have our figure here. So we have SRE1, like upstream. Um, oh, this is the gene over here. And in the, I guess, promoter sequence of that, we have all this other stuff, AP1, blah, blah, blah. Okay. To determine whether LFAB-P selectively influences targeting of LCFAs to nuclei, cultured rat hepatocytes isolated from LFAB-P++ and LFAB-P minus minus rats were incubated with the fluorescent LCFA analog BOTA-P FLC16 and the nuclear DNA binding probe Cyto 61, um, and imaged in real time using laser scanning confocal microscopy, LSCM. Quantitative intensity analysis of the resulting data collected from multiple hepatocytes indicates the relative nuclear and cytoplasmic distribution of BODP within the cells. Okay, let's go down some. All right, figure two, nuclear and cytoplasmic distribution of the fluorescent fatty acid analog in hepatocytes derived from wild type plus plus and minus minus mice. So we see for the plus plus in the nucleus a larger amount for the fluorescence intensity um, compared to minus minus. And then in the cytoplasm, it seems like the other way around, a higher amount of this fluorescence intensity is for the FAB-P minus minus mice. Um, and I think they said that so the FAB-P is inversely proportional to cytosolic LCFAs. So I guess it makes sense that in the cytoplasm, having FAB-P gives you a reduced amount. Um, right?
Mm,
So to determine whether L-FAB-P selectively influences targeting of LCFAs to nuclei, they did this experiment here, um, where they did the fluorescent analog and then the nuclear binding protein cyto61 in real time. And yeah, they looked at the relative nuclear and cytoplasmic distribution of this within the cells. And so for the LFAB-P, like, having that gene um, means that there's a higher amount of it expressed in the nucleus and a lower amount of it expressed in the cytoplasm. So we can probably say, like, so I wrote inversely proportional to cytosolic LCFAs. Maybe I could say proportional to, oops, nuclear LCFAs. And then for the figure beneath that, let's see. Okay. Ratio of this intensities in nucleus slash cytoplasm. So this is so okay, when you have the FAB-P, we have a really much higher amount of this, fluorescence intensity in the nucleus compared to the cytoplasm. So that also makes sense with what we read.
there's more binding. Okay. And again, they're seeing if the LCFAs are, if its ability to bind to LCFAs in the nucleus versus also binding to in the cytoplasm, essentially.
Um, yeah.
Okay.
Okay. So pretty tough passage. Let's try this question out. Passage information and the experimental findings suggest what relationship between LCFAs, PPAR alpha, and L-FAB-P transcriptional regulation. So, what are your thoughts?
Yeah, so when I did my blind review, like going back to this paragraph, like I understood that it seems like PPRE and then PP, oh yeah, you even wrote it here, right? So like when they bind together, they increase the LFAB expression.
Okay.
So, and we know that LCFAs bind to these FABPs.
Yep.
So I'm guessing that as they bind to it, it probably activates it and increases the expression.
Uh-huh.
Um, and then I realized like I was still between A and B, but B says it depends on their synthesis. So I was like, hmm, um,
Okay.
it says it increases expression, so I don't know about that.
Uh-huh.
And
So,
then I don't know if the other two, but yeah.
okay, so we're saying that in paragraph 2, LFAB-P, um, its promoter contains things like the PPRE and the PPAR alpha, and so the PPR AR alpha is a receptor found in the nucleus and can be activated by cellular ligands like LCFAs, and binding of an activated PPAR alpha, the PPRE can increase the L-FAB-P expression and is required for expression of genes involved in peroxisomal and mitochondrial fatty acid beta oxidation and lipolysis. And it also contains these sterol elements that can also play a role. And then they said that to determine whether LFAB-P selectively influences targeting of LCFAs to nuclei, um, they did the experiment. And so they're asking, yeah, from the passage information and experimental findings, what relationship is there between all this stuff? So we wrote in our I wrote in the proportionalities that Fab P causes more LCFAs to be in the nucleus rather than the cytoplasm, right?
Okay.
And that PPAR, when it binds to PPRE, you get more expression of the FAB-P, which means more beta oxidation and lipolysis, but less cytosolic LCFAs and more nuclear LCFAs. So if you look at these choices, let's start with B. B says LFAB-P mediated transport of LCFAs depends on PPAR alpha synthesis. What are your thoughts for that?
Yeah, I don't know about that because again, it says binding has been shown to increase expression. So just that it increases expression, I'm not sure if PPAR is exclusively if FABP transport is dependent on it, if
Mm-hmm.
it helps it.
Mm-hmm. Yeah, because we know that it can behave the way that it behaves even without it. So so yeah, it's something that, let's see. Yeah, this mediated transport seems to depend on PPAR alpha binding to PPRE
Mm,
rather than specifically how much PPAR alpha is made. So I would say that's not something that gives us any information about that relationship. I'm curious to see what they wrote for the explanation. So they wrote, okay, no direct evidence is given to support the L-FAB-P mediated transport of LCFAs as being dependent on PPAR alpha synthesis. Okay. Then C, transcription of the L-FAB-P gene is inhibited by LCFA PPAR alpha binding. Well, we know that when the PPAR alpha binds, that we have more LFAB-P expression, right?
yeah.
So that would be the opposite. C would be the opposite. It should be increased
Mm.
rather than inhibited.
Yeah, definitely. I just I was
Yeah.
running out of time, so I guess.
Well, yeah, there's a lot of stuff, a lot of stuff going on here. So, okay, so now let's see. D says LCFA transport by LFAB-P promotes PPAR alpha mediated degradation in the nucleus. So we know that LCFA transport causes this to be causes LCFAs to be moved to the nucleus, but I don't know if that necessarily means that it's going to be broken down in the nucleus, especially not by PPAR alpha mediated degradation. So that would be my guess for that. Let's see what they wrote in the explanation. The removal occurs in oxidative organelles, um, but there's no indication that LCFAs are degraded in the nucleus. Yeah, there's no information that says that it's degraded in the nucleus. There's just information that says that it moves to from the cytosol to the nucleus, right?
I'm kind of confused as why are they telling us all about like changing it from the cytoplasm to the nucleus, yet they're telling us that it's important they try to take away this toxicity within the cells.
Mhm.
That, for some reason, that's not like clicking in my brain.
Well, they said um that it's toxic when it's in the cytosol,
Okay.
and that you can remove them by putting them in oxidative organelles or the endoplasmic reticulum or and lipid droplets. And then in the second paragraph, they talk about how the LFAB can essentially move uh, the LCFAs to the nucleus,
Hmm.
right? And
Yeah.
that can enhance the peroxisomal or mitochondrial beta oxidation and lipolysis. So we could still break it down in the oxidative organelles, right? But FAB-P itself, what that seems to do is it seems to take the LCFAs and move them to the nucleus,
Hmm.
and that can result in more FABP being expressed or produced.
So basically the else it brings it in to the nucleus, which increases the FABP gene expression.
Uh-huh. Yeah, so
Okay.
FABP itself reduces or I should say, yeah, reduces the, well, I guess I already wrote. So like what I wrote here,
Cytosolic LCFAs, okay.
maybe I'll put it in parentheses toxic. Whoops. So we know that LCFAs at high concentrations in the cytosol is bad, and FABPs can remove the LCFAs, the toxic LCFAs in the cytosol. And it can do that by moving it to some oxidative organelle that will break it down. Or if it moves it to the nucleus, it can help produce even more FABPs.
Mm, okay.
And also, because the FABPs can do like more than one thing. They can break down, cause the breaking down of LCFAs. But
Mhm.
they can also uh ex they can also regulate expression of its own FABP gene.
Mm,
And once it acts to transport LCFAs to the nucleus, it'll cause the expression of the FABP gene to go up.
okay. That makes more sense. Okay.
Yep.
Yeah.
So
That nucleus keeps throwing me off. That makes more sense. Yeah, because it's like, why is it wanting to do that? That makes sense. It wants to increase it.
yeah, yeah. So yeah. So we could say that, I mean, so yeah, for A, we could say so transport of LCFAs to the nucleus activates PPAR alpha. And we do see that here. It's a receptor protein found in the nucleus, and it's activated when bound by cellular ligands like LCFAs, right?
Yeah. Hmm. Hmm.
And so transport of LCFAs to the nucleus activates PPR alpha. That's directly stated here. And when that happens, right, binding of activated PPR alpha to PPRE has been shown to increase expression of FAB-P, right? Which acts to cause an increase in expression of genes that cause this type of beta oxidation and lipolysis, right?
Um, so LFAB is like it's responsible for LCFAs, but it's also
It's just,
responsible for increasing peroxisome and mitochondrial beta oxidation. Okay.
well, the first thing you said, So LCFA, FAB-P removes LCFAs
Mm-hmm.
from the cytosol by, either causing it to move to like a peroxisome or mitochondria to get broken down.
Mm-hmm.
That's what LFAB can do. But LFAB can also regulate its own expression.
Regulate its own expression. Okay.
Meaning, yeah, so like we see that here. So LFAB-P can cause, so we know that FAB-P will reduce the amount of this toxic LCFAs, right?
Mm-hmm.
And it can do that by sending the LCFAs to oxidative organelles,
Mm-hmm.
right? But what it also does is it transports the LCFAs to the nucleus. And when it does that, it activates PPAR alpha, which causes more of expression of FAB-P, which in other words promotes FAB-P synthesis.
Mm.
So
Okay.
that's something that it does to make more FAB-P, and FAB-P ultimately is responsible for the uh, breakdown of fatty acids and lipids.
Breakdown fatty acid and lipids. Okay. That makes a lot
So,
more sense. Okay.
yeah, so here they wrote um, figure 2 shows that the hepatocytes of rats not expressing LFAB-P have decreased nuclear and increased cytoplasmic LCFA concentrations. And we do see that when we don't have FAB-P, the gray,
Mm-hmm.
there's less LCFAs in the nucleus, but more of it in the cytoplasm here, right?
Mm. Yeah, that makes okay. Yeah, that makes okay.
That means that that this means that LFAB-P, if you have it, it'll be the black bars, which means that you should have more LCFAs in the nucleus compared to the cytoplasm.
Then why is this column here bigger than this? Shouldn't this column be like up here and maybe this a little less, right?
I think it's just what's relative, meaning uh
Oh.
yeah, like sure, we still have a higher amount of the LCFAs in the cytoplasm compared to the nucleus, but you want to really I'm saying, and that's for if you have FAB-P, but you really want to compare having FAB-P to not having FAB-P, because
Okay.
that's what these asterisks show us. So basically we see that if you have FAB-P, that you have a higher amount of LCFAs in the nucleus compared to if you don't have FAB-P.
Okay, that makes sense.
And
Okay.
that if you have FAB-P, you have a lower amount in the nucleus compared to if you do not have FAB-P,
Okay.
right? So that's what we see here, that figure 2 shows that the hepatocytes of rats that are not expressing FAB-P have decreased nuclear and increased LCFA concentrations. And that's what
Mm.
we see there. Then it says figure 3 is consistent with this, depicting a decrease in the nuclear cytoplasmic LCFA ratio. So figure 3 just kind of shows us kind of like what figure 2 shows us, but as a direct ratio. So if you scroll down to figure 3, you can see here that this is literally the ratio of nucleus to cytoplasm,
Oh.
that when you have FAB-P, you have a significantly higher ratio of LCFAs in the nucleus compared to the cytoplasm, and compared to the when you don't have FAB-P.
Mm. Okay.
So, so yeah, figure 3 depicts a decrease in nuclear cytoplasmic LCFA ratio. Um, when LFAB-P is there. So this suggests that LFAB-P acts in such a way as to increase the nuclear and decrease the cytosolic concentrations of LCFAs. The passage also states that LFAB-Ps bind LCFAs in the cytosol. Taken together, this suggests that LFAB-Ps serve to transport LCFAs from the cytosol into the nucleus, where they might bind to the nuclear receptor protein PPAR alpha. This in turn binds to and activates PPRE. According to the passage, PPRE increases synthesis of LFAB-P. And that's what they say here. Um, and according to the passage, PPRE increases synthesis of LFAB-P. In this way, LFAB-P may act to upregulate its own transcription. So, tough passage because there's just so much going on, right?
Yeah.
Um, so, but if you wrote out our proportions here, like we did, we could see that FAB-P means less LCFAs in the cytosol,
Hmm.
more LCFAs in the nucleus, and that also PPRE, PPAR alpha, when those bind, causes more FAB-P expression and therefore more peroxisomal and mitochondrial beta oxidation and lipolysis.
Okay.
Mhm.
Cool.
All right. Yep.
In a separate study, researchers identified large vacuoles of abnormal triglyceride accumulation in the liver cells of genetically modified mice. Abnormal triglyceride accumulation in the liver cells. These most likely suggest. Yeah, so when I did my blind review again, I actually picked B, lack a functional LFAB gene. I knew it wasn't overexpression because if they're accumulating, then maybe they're not being like taken out. So either one of those genes probably are an issue. Um,
Okay. So what we wrote in the proportionalities here is that sure, FAB-P can play a role in moving the SCFAs, but it's the PPAR alpha that actually causes the breakdown of LCFAs,
I see what you're saying.
right? Because we said that um yeah, that PPAR alpha is a receptor protein found in the nucleus that is activated when bound to things like LCFAs. Binding of activated PPAR alpha to PPRE increases expression of FAB-P and is required for the expression of genes involved in peroxisomal and mitochondrial beta oxidation and lipolysis, right? So
Yeah.
what I wrote here was PPRE binding to PPAR alpha causes more FAB-P expression, sure, but it also causes more more beta oxidation and lipolysis, meaning breaking down fats and fatty acids.
Okay. That makes sense.
So if we saw an accumulation of triglycerides, it means that we're having an issue with breaking down fats, right?
Yeah, because they could still be like being taken in,
Mm-hmm.
still not being broken down.
Yep.
Damn, okay.
So so we know it's not going to be Fab P because, well, Fab P should cause, perhaps more um degradation of the triglyceride because it causes more of the PPAR thing.
Yeah.
Uh, but the PPAR thing is what directly can cause, the breakdown of fats. So, yeah, it's most likely going to lack a functional PPAR alpha gene. So the passage states that binding of activated PPAR alpha to PPRE is required for the expression of genes involved in peroxisomal and mitochondrial beta oxidation and lipolysis. So if you lack that gene, this type,
Hmm.
this type of binding of PPAR alpha to PPRE will not occur. And if it doesn't occur, there's not going to be that type of lipolysis. So you'll see an accumulation of triglycerides.
Okay.
So
That's good.
it'd be a bit confusing. I could see that great more than 50% chose lack of functional FAB-P gene. But it seems like the FAB-P gene can perhaps, enhance this type of lipolysis, but it only does so by affecting the PPAR alpha. It doesn't directly do anything like that.
Okay. Alpha. Okay, so based on the information present in the passage, the alpha promoted oxidative breakdown occurs in which organ? No. Yeah, I don't know how I missed this, but yeah, I guess it makes sense. So it's mitochondria and peroxisomes, yeah, because again, it's the acquired expression of genes involved in peroxisomal and mitochondrial fatty acid oxidation.
Uh-huh.
Um, so yeah, I see why this is one and two. This makes sense.
Wait, show me where you saw that answer?
Increased expression of LFAB, right, and is required for expression of genes involved in peroxisomal and the mitochondria, right? Because we said the PPAR to PPRE activates that. Well, it's activated by LFAB, and then also activated. It also expresses these peroxisomal and mitochondrial beta oxidation.
Yeah, so I think that's telling us more so about how the PPAR alpha can do this. But for this question that's asking about LFAB doing it, it's more so this part, FAB-Ps, directly rapid removal in oxidative organelles. I think that's what they mean.
Okay. Yeah, I think also when I look back on it, that's a sentence that I focused on too. But when I read oxidative, I just figured it was just mitochondria. Like for some reason, the thought of the peroxisomes, like I guess in the moment I think I forgot the function of it. So maybe that could be it too. But yeah, okay. A mutant alpha protein with greatly reduced LCFA binding affinity differs from wild-type LCFA at a single critical amino acid residue. Which of the following substitutions is most likely to account for this change in LFAB LCFA binding?
Okay. So we want to see like okay, so we're used to at this point seeing like, active site amino acids being changed into a different type of amino acid and then what type of substitution would, cause the greatest amount of change. So if we're looking at this, for instance, well, let's see. So lysine to threonine, um, well, oh wait, hold on. The mutant LFAB protein, a mutant LFAB-P protein with greatly reduced LCFA binding affinity occurs or differs from wild type LCFA at a critical amino acid residue. So they said that the FAB-Ps bind to the anionic component of LCFAs, right?
Yeah.
So FAB-Ps would have what type of amino acids.
Yeah, probably something that's positively charged so I can bind to it.
Correct. So for instance, so for A, we have positive lysine to, neutral threonine. B, we have negative aspartic acid turn into, serine, neutral serine. Then C, the glutamic acid, negative to aspartic acid, negative. And then valine, neutral to neutral, right? So the only one that, you know, could bind to anionic stuff would be the lysine. And
Mhm.
if you got rid of that because it has a positive charge, and if you got rid of that positive charge, it's not going to bind to the LCFAs very well.
Yeah.
So
Yeah, I think I again I think I missed the and it's just a small little thing like this all this stuff just
yeah,
threw me off the little anionic portion.
yep, yep,
Because yeah, oh that
yep.
makes a lot more sense. Awesome.
Fortunately, it seems like, when I first saw this passage, I saw figure one just looks kind of like a mess and like, you know, and then when they asked us about SRE, oh, go back up.
Hmm.
SRE over here, like, we kind of didn't really think about that too much. Um, but you
Yeah.
see how the questions that they ask us, like, didn't even require us to know about, like, to even use figure one. So a lot of times, like, really kind of complicated looking things like that. Like, so when you're doing your test, when you analyze when you do the data interpretation, I would say spend no more than 15 seconds trying to see what it's trying to tell you. Um, if it takes longer than 15 seconds, just move on to the questions. And if the questions ask you about, going back to one of the figures, then you can go back to one of the figures when that question asks you. But aside from that, yeah, don't spend more than like 15 seconds because, like, either they'll ask you about it, in which case you can just go back to the passage and look at the figures that way, or they won't ask you about it, in which case you don't have to ever even look at the figures. So I think figure one here is a good example of something that wouldn't have made sense for us to spend too much time on when we first analyzed the passage. Um, and therefore it's better for us to not really spend too much time on something like that, unless they ask us about it.
Okay.
Because they
Cool.
didn't really ask us about figure one, right?
Yeah, barely nothing.
For the sterile, element, SRE1 and stuff.
Yeah.
So, yeah.
Interesting. Okay. Um, I guess we can also do this. Um, but I haven't really attempted this, to be honest, because it was another thing that I ran out of time. Do you suggest that we do something that I haven't, that I have attempted, or
Yeah.
pass
Hmm.
something that I've attempted?
Yeah, blind review.
Okay, let me see.
We were
Okay. Um, no, this makes sense. Okay, what are we gonna do? Okay, let's do this. All right. No, no. Okay. All right.
Sorry, I went to get more coffee.
That's a lot of coffee, man.
Yep. Okay, so this is one that you did your blind review on.
Yeah, and I still got 41, 42. I think I only got this question right, which was pretty easy, but yeah, the rest of it was just odd. I kind of understood even after I took it, but I don't know. I think maybe we should just go over it.
Okay, let's see. Epigenetic mutations can stem from modification of germline epigenetic programming during the gonadal sex determination period when germline DNA is demethylated and remethylated in a sex-specific manner. What uh what would methylation of DNA do?
Yes, I was confused when I was reading that. I know if it's methylated, I think that blocks gene expression, but if it's demethylated, that should open up gene expression, right?
Yep.
Okay.
Modified epigenetic programming in males leads to all tissues propagated from the sperm to have differentially altered epigenomes and transcriptomes that can influence development of adult onset disease. The altered epigenome is transmitted to subsequent generations due to apparently permanent imprint-like DNA methylation properties. Um, Certain environmental chemicals are known to promote this epigenetic inheritance. Okay, so they're talking about how epigenetics can play a role in some disease starting once you're an adult. Um, okay, let's see. The epigenetic actions of and then environmental chemicals can cause this epigenetic inheritance. So compounds BPA, DEHP, DBP were investigated. These compounds were selected because they are endocrine disruptors that are derived from household plastics. It was postulated that the exposure of a gestating female rat, F0, to plastics would promote epigenetic inheritance. Tests for diseases of the testes, prostate, kidney, ovary, tumor development, and obesity were performed on 1-year-old rats from the F1 and F3 generations. Okay, scientists predicted that the phenotypes observed in the F1 and F3 generation rates would be similar, but have different causes if epigenetic inheritance were to occur. Okay. So I guess here what they're saying is that they did experiments with these household plastics, um, and exposing gestating female rats to these plastics can cause this epigenetic to see if it would cause this epigenetic inheritance. And they did these tests for the F1 and F3 generations, and scientists predicted that the phenotypes that you observe in the F1 and F3 generation, the rates of the phenotypes would be similar, but have different causes if epigenetic inheritance were to occur. Okay, three groups of rats with varying levels of plastic exposure were used. Control, low dose LD, and full dose plastics. Figure 1 shows the incidence of select diseases and tumor development in the F1 and F3 generations. Okay, let's look at that. Okay, figure 1, selected disease outcomes in the F1 and F3 generation of rats. So let's see. A shows, kind of hard to see, but let's see. Percent, percent pubertal abnormality. F1, and this is for females, okay? F1 control, super low. F1 plastics, super high. F1 LD plastic, that's I think the low dose one, right?
Yeah, I think yeah, those.
Okay. It has a smaller amount, um, but greater than control, of course. And then F3 control, small amount. F3 plastics, largest amount. LD plastics, smaller amount. So maybe I would say something like abnormality proportional to the, I guess just like plastics, amount of plastics. And then for male, uh, for male, for F1 male, it seems like the LD produces a larger amount of abnormality than the full plastics. And then for the F3 control, it's what we more along the lines of what we expect, with the plastics being the highest compared to LD plastics. So, okay, so we get to see that for the female F1, but it seems like it differs for the male. And then for the follicle loss, um, seems to go up more with the LD. Okay. And then for polycystic ovary disease, seems to go up with the LD as well. Female tumor development, um, for F1 seems to be highest for control. F2 highest for plastics. Male tumor development seems to be almost non-existent for F1 except for LD, and then goes up from control to plastic and then to LD for that for the male. So we have various different, I guess, kind of values that we see here relative to, what we're looking at. So a lot of different kind of things. So, okay, let's see. Um, Figure 2 shows the total disease incidence in males and females in the F1 and F3 generations, as well as the incidence rates of multiple diseases in male and female rats. So let's see. Total female disease incidence, highest for plastics, not the LD one. Total male, highest for plastics for F1, but for F3 seems to be higher for the LD. Then incidence of female multiple disease seems to be highest for F1. Sorry, for highest for both to be plastics. for the male multiple diseases, highest for F1 plastics, but for so it seems like LD plastics. Whoops. For the males, I guess at least for F3. So, okay, so what we got. So, all right, let's try these. Um, the researchers did not present data from the F2 generation. What is the most likely explanation for this? Um, okay, let's scroll up on the left. They might have said something like that. So they looked at the F0, which is gestating female rat, and they used um, the tests on 1-year-old rats of the F1 and F3 generations, and they predicted that these phenotypes would be similar but have different causes. Um, okay, and we, I guess, just have to figure out why um, they used F1 and F3 generations rather than the F2 generation, and nothing in the passage seems to tell us why. So I guess, we're gonna have to like whatever, see what could be a reason for this. The data from the F3 generation mice showed more statistically significant discrepancies with the F1 generation. Um, there's I don't think that would explain it because I guess if they're saying that the F3 generation mice showed more statistically significant discrepancies with the F1 generation compared to the F2 generation, we don't really have any any information that tells us
Mm.
anything about F2 generation to figure out if, it's more, if F3 or F2 is a more significant
Mm.
discrepancy. I think that would be my guess for that. B, not enough time had elapsed by the F2 generation to account for the possibility of spontaneous reversal of endocrine disruptor induced methylation. I don't really see that making sense because if not enough time had elapsed by the F2 generation, then why would, I guess,
Spontaneous reverse.
F1. Yeah, because F1 should happen before F2. So
Mm.
if they're saying not enough time passed by the F2 generation, then why use the F1 generation? My guess would be. Uh,
They're not even checking for spontaneous reversal, right? They want to see the effects of how this endocrine messes with the methylation, right?
yeah.
Okay.
So I think it's this type of um this thing, I guess, sex specific manner. Um, but okay. C, epigenetic mutations are recessive and therefore skip generations. So epigenetics is like, what is epigenetics?
Um, I know it's like
Mhm.
changes in DNA, right? Which can affect gene expression, I think. Something about changes in DNA. That's all I kind of know.
So, uh, so the so epigenetics essentially means that your genes don't tell you the full story. Like it's not like just your DNA that uh, determines whether you have a disease or not. It's whether that DNA or parts of that DNA is expressed or not expressed, which
Hmm.
means that something like you might have the genes for some disease, but if that gene is not expressed, then you don't exhibit that disease. But if the gene is expressed, you might exhibit that disease. And it's therefore caused by non-genetic mechanisms. For instance, like methylation can turn off gene expression. Acetylation can turn on gene expression. So epigenetics are not changing anything in the DNA, but it just changes what in the DNA gets expressed, meaning what in the DNA gets transcribed into RNA and then translated into uh proteins.
Okay, so it's like you're not like mutating or anything. You're
Correct.
basically like opening it up and it can do whatever it wants, basically.
Yeah.
Or anything can happen to it.
Yeah. So you can either um yeah, like open up like so like between the histones is like the linker DNA, and that DNA can be if it's um if the space between the histones increases, it exposes more of the DNA to polymerases and or transcription factors rather.
Mhm.
That can cause transcription of that gene. But you can also have methylation, which will cause the DNA to condense, and the exposed DNA between the histones will be less exposed, therefore less transcription factors would have a harder time in binding to that DNA to cause transcription of that gene. So, yeah, so you're not changing anything in the DNA itself. So,
Mhm.
we can't say that epigenetic mutations are recessive,
Mhm.
right? Because it's not something that, it's not a Mendelian um, inheritance pattern.
Okay. Then when would it like let's say in another aspect, like when would this answer choice make sense? It would only be if the passage stated that like there was some like typically they would state it, right? Like, oh, like this gene is something that's recessive, right?
Yeah, yeah, yeah. So like if they're talking about actual like Mendelian inheritance where they can give you like a pedigree
Yeah.
or they can tell you something about like uh how it like if like let's say that they give you a pedigree and something like skips generations or something like that, that would be more along the lines of something that you would see. But those are literal mutations of the DNA.
Yeah.
But yeah,
Mhm.
epigenetics don't really change the DNA.
Okay, that makes sense.
Yep. And so then D says the fetal germline cells of the F2 generation were directly exposed to the endocrine disruptors, making it difficult to distinguish between the effects of direct exposure and epigenetic inheritance. So they told us that they were investigating the epigenetic actions of these plastics, and they were selected because they are endocrine receptors, or sorry, endocrine disruptors, and that if the a gestating female rat was exposed to this, it
Mhm.
can cause these type of diseases in the F1 and F3 generations, and that scientists predicted that these phenotypes in the F1 and F3 generations would be similar, but would have different causes if the epigenetic inheritance were to occur. So
Okay.
what is the most likely reason for them not giving us F2 generation info? Well, if it was the case that they're exposing the F2 generation, the fetal germline cells of the F2 generation to the endocrine disruptors, we would not know um, if the effects of what we see is due to. So, okay, I should say this, that they take the F0 generation and they treat that with the plastics, the endocrine disruptors, and then we can see in the F2, I'm sorry, the F1 and F3 generation, the effects of epigenetic inheritance. But
So,
um, but that means that we're exposing the F0 to the endocrine disruptors. And then
mhm.
when we look at F1, we can say that the things that we see in the F1 generation um, is due to not being exposed to plastics, but inheriting something from the F0 generation that is exposed to plastics.
Right.
So that means that so F1 can show the inheritance because we exposed F0 to plastics. Now, how can we say that F3 is the product of inheritance? That must mean that in the F2 generation, we exposed that to plastics too, which means that the F3 generation would inherit the epigenetic, effects from that that was directly added to the F2 generation.
Whoa, okay. Taking that in. Okay, so when you're basically saying that again, yeah, so F0 was exposed with the plastic, so now you get the F1, but you had to give plastics again to the F2 because not all of the F1 might have been affected by the plastics, essentially.
Well, if we're trying to figure out um, epigenetic inheritance, we would want to expose like one generation to the endocrine disruptors, aka the plastics,
Hmm.
and then look at the next generation to see if we have epigenetic inheritance.
Okay.
So that makes sense in that for the F2, F0 generation, we expose them to the plastics. So the F1 generation that comes from the F0 generation will show epigenetic inheritance, right?
Yeah, sure. F1, yeah.
Now, they're asking why are they giving us data for F1 and F3 but not F2? And it could be that F2, like you can't go from like uh an epigenetic inheritance like from F0 to F1. You can't go from F1 to F2 with another level of epigenetic inheritance. It must be like for F2, it seems like you can only go down like one generation. So either we expose F0 to plastics, evaluate F1 for epigenetic inheritance, and then we expose F2 to plastics to evaluate the effect of epigenetic inheritance in F3, right? Rather
Mhm.
than just exposing F0 to plastics and then looking at the epigenetic inheritance for F1 and then F3. It doesn't seem like it'll work that way. Because you want to evaluate the generation that comes after whichever generation you directly add the endocrine disruptors to.
Okay. No, it makes sense, but it feels like it's a little bit of like
Hmm.
outside passage info, but then again, it's like the last three answer choices too, they're pretty wrong. So
Huh?
that
Yeah.
makes I see what you're saying. So I understand what you're saying, but it's again like it's not in the passage, so it's weird for me to.
Yeah. So I think that, yeah, because they're asking just what's the most likely explanation for this. So, so let me see what they wrote. So they wrote the gestating F0 mothers who were exposed to the disruptive plastics mixture contained fetuses who became F1. The germ cells contained in those fetuses became the germline cells of the F2 mice. Therefore, the germline cells of the F2 mice were directly exposed to the endocrine disruptors, making it challenging to distinguish between the effects of direct exposure, as was clearly the case for F1, and the effects of epigenetic inheritance, as was the case for the F3 mice. So I think that they're telling us that the F0 mothers were exposed to the plastics and they had fetuses that became F1,
Hmm.
and the F1 fetuses contained germline cells that became F2, which would mean that you're exposing the F2 to the endocrine disruptors, the plastics,
Yeah.
and therefore it's hard to know if the effects of that is due to direct exposure of the plastics or epigenetic inheritance.
Okay.
Mhm.
Cool. Most likely that BPA, DEHP, and DPB interfere with endocrine function by interacting with. Yeah, so I picked membrane-bound receptors because I figured it's endocrine, so it's got to be like I'm thinking maybe like insulin or like all that stuff,
Well, they're
right?
talking about, oh, sorry. So they're talking about in the, you know, the types of issues that they see are like female, like after puberty, right?
Hmm.
If we scroll down on the left, we could see, so A and B, like in figure one, A and B shows female pubertal abnormality and male pubertal abnormality. So something's happening at the puberty stage. Like the abnormality is being expressed at the stage of puberty, right?
Yeah.
And then we see primordial follicle loss, which could be related to like follicle stimulating hormone kind of deal, and polycystic ovary disease. Um, so all of these things involve um, things that happen during puberty. And what type of hormones cause changes during puberty?
Estrogen,
Mhm.
progesterone, testosterone.
And what type of um, hormones are those?
Yes, they're typically steroids, right? Because they go into the nucleus and they
Yeah,
mess with binding. So,
yeah, they're steroids. So basically, I mean, for this question, we can get rid of A and D, right?
hmm.
Um, and then it's just whether it's a non-steroidal hormone, like a peptide hormone, which would require a membrane-bound receptor for it to do its work, or a steroid hormone, which, is non-polar and because it consists of those rings and can simply bypass the lipid bilayer of the cell
Hmm.
membrane. It can also bypass, the nuclear envelope or nuclear membrane to directly cause some type of change in transcription at the DNA level. So yeah, they're asking what these types of plastics like. Well, we know that they disrupt endocrine function, but how? Yeah, like how do they do that? Like with what endocrine hormones, right? And
Yeah.
we see that it's predominantly puberty type of stuff. We think, okay, what type of hormone plays a role in puberty? And it's like estrogen, testosterone, and those are all, you know, uh, things that interact with the nuclear receptors because they're
Oh,
steroids, right?
yeah, yeah, that makes sense. Yeah, I just missed that. I just clicked thinking that endocrine, but yeah, I
Yep.
didn't know. Okay.
Hmm.
All right. Yeah, this was easy.
Yeah.
Different derivative hormones.
Hmm.
It's funny, I got this right.
Yep.
Which of the following conclusions is most supported from the findings of the study?
Okay. So I see what they wrote for C, which is kind of tricky because, yeah, for figure one, A, B, C, and D all have asterisks, right?
A, B, C, and D. Yeah.
But E and F has have no asterisks.
Oh, okay. Did not notice that, but now I notice it. Okay.
Yeah, yeah. So, and E and F are talking about, female and male tumor development, which could play a role in oncogenesis, like cancer, right? But yeah,
Hmm.
no statistical significance being shown there. So
Yeah.
you can't say that C is true, right?
Yep.
But given that the F3 generation had no direct exposure to the endocrine disruptors, yet we see that for A, B, C, and D, um, meaning figure one, A, B, and C and D, we see that F3 definitely has statistically significant, you know, effects. And the F3 generation did not have direct exposure to the plastics, aka the endocrine disruptors. So, yeah, we could say that it's not necessary to have direct exposure in order to have endocrine disruptors affect your health.
Okay, that makes sense. And
Hmm.
then why would, okay, so A says endocrine disruptors shows a consistent dose-dependent effect. Okay, yeah, I see that because like I mean, we see a little bit with the female pubertal, but like it's hard to say because sometimes low doses do more and then
Yeah,
sometimes doses do more.
yeah, yeah. That's something that we observed first, right? We saw like, because at first I wrote like, what did
Yeah,
I,
pubertal, right? I think that's this. No, abnormality. That says abnormalities.
yeah, abnormality proportional to amount of
Plus,
plastics. And then I had to change it to be like female F1. But yeah, that's like I couldn't really write anything else for that because we didn't see any type of, uh,
no.
proportionality relationship like that.
Okay.
Oh, yeah. So it
And
won't be a dose dependent effect.
then D says epigenetic inheritance weakens the negative effects of endocrine disruptors. Uh, yeah, I'm not sure about that. But yeah, the values are like all over the place, right? I think that's a little hard to assess, right?
Yeah,
Mm.
right. Because then we would have to look at epigenetic inheritance like F1 compared to, you know,
F3. Yeah, that'd be hard to tell because all of these values are all over the place.
Mhm.
That makes sense. Cool. Yeah, I think that was really it for BB. Everything else I kind of understood, but yeah, that's like the biggest thing that I'm noticing. It's yeah, the passage analysis, I guess. But
Mhm.
what do you think so far with what I've shown you?
Yeah, no, I mean, like these are really tough passages, and that's kind of like the idea behind doing these blueprint tests. So,
Okay.
so yeah, I think that you're doing a good job with your results. But yeah, we're definitely going to work on that timing.
Okay. Oh, and then I have a couple questions, CP. I have you for 20 minutes, right? 3:15, right? Until 3:15?
Mhm.
Okay. Yeah, so for cars, I wanted to quickly go over that then. Like, what should I do? 'Cause I asked you earlier, but yeah, we just moved on to this. But yeah, what do you think I should do? Like, should I set a goal for myself to try and get to 6, 7? Um, my plan is like starting tomorrow, I'm gonna try to make sure I'm getting at least like I'm finishing a passage under 15 minutes
Uh-huh.
more,
Yeah,
but yeah, I don't know, like yeah, sorry.
yeah. So yeah, I think the 1.5 minute thing is gonna help. But yeah, because it seems like the timing seems like something that you could benefit, like that you can work on for like essentially all the sections besides psych soc. So um yeah, so I would say that basically the same thing as the science sections in terms of the timing. But yeah, if you want, you can with your current practice of cars, you can kind of experiment with doing you know, I guess in your practice, yeah, to experiment doing it timed. Because I guess I said like not to do anything timed except for the practice tests, but since you're at the point where the timing is a main issue, then for the practice that you do, you can do it timed. You can kind of maybe try to do um like a passage in like let's say like 10 minutes. We can aim for that. So like let's say that you do like two like let's say like tomorrow or whatever you do like 2 to 3 cars passages. Um try to do them in like let's say 10 minutes. And
Okay. Yeah.
so now since we're doing it under a time condition, that means that blind reviews will also happen. So basically you take a cars passage, you try it uh with with a 10-minute like timing constraint, and then you see how you do, and then write down which questions you got wrong, and then do a blind review after where you take your time doing that cars passage, and that can be the workflow from now on. You could do your cars passage in 10 minutes timed, and then after that you do your blind review.
Okay. With that in mind, then what should I, because I mean it's exam.
Oh, you froze for me. Hello?
Always a Wi-Fi issue here, bro.
Huh?
Hello.
Okay.
Can you hear me?
Yeah, I can hear you.
Okay. I was good. I was a little scared. I was like, God damn it, again, the Wi-Fi. Um, yeah, so exam crackers is kind of an issue because you have to go to the end of the thing and like it tells you the answer. So I wouldn't know if I got it right or wrong. But yeah, what should I what should I do at that point then?
What was your question?
Yeah, so because I see what you're saying in the blind doing a blind review for cars, but I would need when I'm doing exam crackers and I do a passage, I have to go all the way down to figure out if I got the question wrong. But like it tells me, oh no, it's B or C. So like how would I? Because with Jack Weston, I can just press submit and it tells me how many I got wrong.
Hmm? Got it. Um, well, I mean, yeah, it'll tell you like I guess what the right answer is, but you know, I'm trying to think like okay, like if you look at it and tells you the right answer, like what are the chances that you'll like, remember it like right away? Let's see. Yeah, I guess I mean I guess it'll be fine, um when you do your passage and then you grade it. you can, circle which ones you got wrong. And like, sure, if you see that they tell you the right answer, like it's not going to be too big of a deal. Um, when you do your blind review, like sure, if you remember it. Because like if you're looking at if you're like write down your answers for like 7 questions and then you look at the right answers, I don't know how it'll be if it'll be necessarily realistic for you to like particularly remember. Like if you wrote like A, B, C, D, E, whatever for questions 1 through 7, and you look at the answer and you just write down which ones you got wrong, like put an X next to it. I don't think it'll be. I don't think like it'll be something that you like memorize right away because it'll just show you the answer in terms of A, B, C, and D. So.
Okay, I see what you're saying. Okay, okay. And I'm also gonna do them in bulks, so I'll probably forget it too. Okay, that makes sense. Okay.
Yeah, that could work too also. Like, yeah, if you do 2, 3 passages and then you grade it and look at the answers, it'll, you know, be harder for you to kind of remember that because you're not gonna memorize like, the answers for like 3 passages. And, even if you do for some of them, like, it's not a big deal. It's still gonna be worth it for you to do that blind review.
Okay, sounds good. Um, yeah, so I wanted to go through this question
Okay.
specifically. So short chain, so that's from the CP section. Short chain saturated fatty acids present in some animal products can produce a rancid odor if they're released from a triglyceride. Which of the following would prevent this type of rancidity? And essentially here they're talking about like um what is it? Food waste is an issue, and the way that they do is like they put preservatives to reduce this with this BHT, and apparently it's an antioxidant that's common in unsaturated fat to prevent oxidation, which leads to rancidity. But
Let me see. Food waste is a major problem worldwide. 1 out of every 4 calories produced by agriculture is not consumed. For the food industry, maximizing shelf life is a paramount concern to reduce food spoilage while en route to the consumer. Consequently, preservatives such as BHT, figure 1, are often added to food products. BHT is an antioxidant that is commonly added to foods containing unsaturated fat to prevent oxidation, which leads to rancidity. So oxidation is going to cause rancidity, right? So short chain saturated fatty acids present in some animal products can produce a rancid odor if they are released from a triglyceride. Okay. Which of the following methods would prevent this type of rancidity? So, so we know that these fatty acids would well, the triglyceride is the bonding of the fatty acids to glycerol, right?
mm,
So, um, so we want and so the short chain fatty acids themselves appear to be what causes the rancid odor, right? So in order to prevent this type of rancidity, we don't want the triglyceride to be broken down into glycerol and fatty acids, right?
mm,
So that's what we want as our goal. So that means that um, we don't want the breaking down of a triglyceride. And the breaking down of a triglyceride is going to be through hydrolysis, right?
yeah.
So we don't want that. What we do want is the formation of a triglyceride, and that happens through dehydration synthesis, right?
Yeah, formation, yeah.
So we want Roman numeral 2. Um,
Oh, so that's what it means, dehydration. Okay. Okay,
uh-huh.
yeah, I didn't, yeah, I didn't understand. That makes sense. Okay.
Yep. Now, okay. Now, hydrogenation is not hydrolysis, but it's essentially what you do for like, if you have a double bond, oops, like a double bond, and you hydrogenate it, you basically get rid of the double bond and you replace it with hydrogens. That's
Okay.
what hydrogenation does. So that can take, that can get rid of a um well, this type of fatty acid is saturated or unsaturated.
Unsaturated.
Yep. So you could hydrogenate an unsaturated fatty acid to get rid of that double bond to make it saturated, right?
Mm-hmm. Yeah.
So they're asking about short chain saturated fatty acids. The fatty acids are already saturated,
Yeah.
so hydrogenation should not do anything then, right?
Mm-hmm. Yeah.
So Roman numeral one is out. Now, sterilization, they perhaps talked about that in the passage or something. Let's see.
I don't think so. This is the end to it.
Let's see. Okay, let's go back up. So, okay. So there could be bacteria that function to break down fats, and that if you remove the bacteria through sterilization, that you can slow down that type of hydrolysis. Bacteria often have lipases that catalyze the hydrolysis of the ester linkage. Um, yeah. So
Is that something that it's like that's outside knowledge, right? So I would need to just know that.
yeah, I guess, what can lead us to thinking of sterilization? I guess you can think like maybe so okay, like it would be outside knowledge to know that bacteria have these lipases, but maybe just from like common knowledge, you can think, hey, sterilization should prevent like something from some food to become rancid, right?
Yeah.
So that could maybe be, you know, something that can clue you in. Uh, but I can totally see why um B had the highest percent of people choosing it because we know for a fact that Roman numeral two is something that we want, and we know for a fact Roman numeral one is something that we don't want. So we
Yep.
can get rid of A, we can get rid of C, and then it's between B and D, and then yeah, it's outside info, I guess. To I would say it's outside info to know that bacteria have lipases,
Yeah.
but it could be more common knowledge to just know that if you sterilize something, it's probably gonna prevent it from like a rant from something from to become rancid. So
Sounds
that could
good.
be something there.
Make a note of it. So yeah, I understood all these questions. We can go over last time. Um, there was one more. I'll find it. Oh, actually, we can. I wanted to do this.
Oh, okay. Um, a woman is standing 2 meters away from a speaker. Um, the speaker is turned up to 11 on its dial, and the power of the sound hitting her tympanic membrane increases 100-fold. Um, how far would the woman need to move to reduce the sound back to its original decibel level? Okay, so the decibel formula is, is this. So we can see that it's definitely related to its intensity.
Hmm.
And we should also know that intensity is power over area, right?
It's essentially the amount of energy the sound wave makes sound or whatever over a certain area, right? Over a certain length, maybe
So power will be the energy uh per time.
energy per time.
Yeah, so
Hmm.
it'll be the energy per time uh per area. And if they're telling us that the power increases 100-fold, then we should have the intensity increase by 100-fold, right?
Okay.
Now, so let's say that increases 100-fold means that, let's say, maybe I'll say like maybe delta I would be 100 or 1E2. Um, now the other thing here is that the further you get from, let's say, like a sound source or something, like uh so okay, since this is also inversely proportional to area, the further you get from that source of sound, um, let's say you get further away from it by 1 meter, the
Mhm.
intensity of that sound decreases by like a square of that distance.
Yeah. Yeah, I think it's what I over 1 over R squared or something, right?
Mhm.
Yeah.
So, yeah. So basically, if we are trying to, so okay, if we know that the intensity increases by a factor of 100, the and we know that the radius squared is proportional to the area,
Hmm.
and the radius is how far you are from that source of sound. In order for you to create an area difference of 100, the length or the distance should increase by a factor of 10.
Can you say that part one more time? I'm sorry.
Yep. So, um, so for every, so like let's say this is the source of sound, and this is 1 meter away from that sound source. It means that the area increases by 1 meter squared, one
Oh,
square meter. Um, let's say that the distance is now 2 meters. The area will increase by a factor of 4 square meters, right?
okay.
So we want to see how far you have to be with this inverse square relationship that will return uh that intensity back to its original level. So if we increase the intensity by 100,
Hmm.
How far must you move away from that sound source in order to have um the intensity dropped by 100?
So probably by the same thing, right? Because if you increase intensity by 100, you probably have to
Mm-hmm.
the area by 100, right?
Uh-huh. So now since area is proportional to radius squared, and we want to decrease that area by 100, we would just have to decrease, or rather, increase that radius by the square root of 100,
Which is 10.
which is 10, right?
Okay.
So if we are saying that the person originally is 2 meters away from the speaker, right? We can say that she needs to be like uh her if her original distance is the 2 meters, the new distance must be 10 meters on top of that,
Okay.
right? So so that's so if yeah, so if the original distance is 2 meters away, it'll have to be 2 meters times 10 meters to give you 20 meters away from that speaker,
Okay.
right? So that's going to lead to um, a movement of 18 meters
Mm.
from the 2 meter point.
And
So,
why are we not adding or anything? It's just why is it multiplication?
oh yeah, it's multiplication because we know that this is going to change. The radius is going to change by a factor of 10, right?
Okay. Yeah.
So whatever that original distance was, we're changing that by a factor of 10 to be 20 meters. And then it's asking how far would the woman need to move. So
Mm.
we start with 2, and from 2 meters to get to 20 meters.
It's just 18.
Yeah.
Okay.
But you can see how most people chose C because that's what you get in your calculation for the 20 meters. And that would be the answer if they asked how far would the woman need to be to reduce the sound back to its original decibel level. But it's really asking how far would the woman need to move.
Okay.
So that's what will lead you to the 18-meter part. But the main lesson here is that things um so you could even do like an experiment with uh let's say your internet speed. Like let's say you use your phone and you're right at where your router is,
Hmm.
like, and you connect to Wi-Fi and you do your and just Google internet speed test, you'll get a certain speed. And then let's have you move, like, let's say 1 meter away from that router and then check your speed. You should see it decreasing by uh like you should see your new intensity or new speed being something that's smaller by like r squared essentially.
Okay.
Then let's say that you move like 2 meters away, or you can basically do an experiment where you measure the speed 1 meter away, 0 meters away, 2 meters away, 3 meters away, and you should see an inverse square relationship.
North square relationship.
Just
Okay.
because as something emanates from its source in all, different directions, it'll be an inverse square relationship. Yeah.
Cool. Yeah,
Yep.
that was it. Thank you so much.
Yep.
One real quick. Give me a second. So any closing thoughts or things I should any suggestions? What do you think? Anything else?
Just that timing thing and practicing with the timing as well.
Okay. Anything I should also do that with BB and CP too when I practice?
Um, yeah, yeah, let's do it for that too. Yeah.
Okay.
That way we can, yeah, we can do a lot of fix a lot of the timing stuff like before your like next test, for instance.
Okay. You still scheduled to take it this Saturday?
Yeah.
Okay. How much is it gonna be?
Let's see. 173.